So, welcome everybody on the first webinar in the grant period 3. It's already third grant period, and we will start with the presentations about the STSMs which took place on a second grant period. And our first presenter will be Angela Balzano. The floor is yours.
Okay, thank you for the introduction, and thank you for this opportunity to have to get this grant and to have the possibility to be involved in this work. And thanks to this short scientific term mission, I could participate to a review that is going to be published soon, and is it supported the goals of VP2 and VP5 of this cost action. And it's about tannins characterization, and I worked mostly on the part of microscopical characterization of tannins, and I reviewed all the methodologies about the identification and characterization of tannins with microscopic techniques. I am Angela Balzano, and I work at the Faculty of Biotechnical Faculty in the Department of Wood Technology. And we work mostly with wood anatomy, combining both the analysis on wood as a material from also archaeological material and also biological tissue. I was hosted by Professor Veronica D'Amico at the Faculty of Agraria, the Department of Federico II, and we collaborate already long time on Mediterranean species and their characterization. So the Professor Veronica D'Amico is a botanist, and she's very involved with biological tissue and also identification of tannins. Tannins identification requires very precise methodology to characterize them at tissue levels. And as I told you, the aim of my short-term scientific mission was to review all the different microscopy methodology that that can allow the identification and the characterization of tannins. I reviewed firstly the techniques for optical microscopy with the methodology that are already used long time ago, such as the protocols with the vanillin or ferric chloride tests. And those methods are quite effective and simple to apply, but sometimes we need higher technologies. For this, then the second part of the review was to identify the methodology used with the fluorescence microscopy. So also all the different staining techniques, and also to the techniques that use the autofluorescence of tannins. This advanced application combine also confocal spectral imaging, cryofluorescence, and mostly those techniques are applied to lignified tissue or plant tissue with the high spatial resolution. And additionally, also I searched for other technologies like confocal Raman microscopy, which is quite specific for chemistry identification of the tannins. And so it's quite also it's quite not destructive method. This is something that sometimes is of high importance, especially if we speak about archaeological remains foundings. And in the end, I reviewed the methods regarding scanning electron microscopy as combined with the X-ray spectroscopy, since the combination of these two can give both a topographic view of tannins, but also the in situ chemical characterization, thanks to the energy dispersive X-ray spectroscopy. So in conclusion, I compared the all strengths and limitation of each approach, and I structured this founding in one paragraph, which will be a part of the review, which is going to be soon submitted. And this work helped the goals of BP2 and BP5 of the cost action. And we are waiting now for the final review of the manuscript, which then will be soon submitted. So yes, I would like to thank you again for this opportunity.
Thank you.
Thank you.
And are there any questions? If no.
Yes, I have a question. Hello.
Yes.
Angela, when you speak about archaeological wood, what's the preserved condition? Because we have a lot of waterlogged wood, and I would be interested if this is applicable also to waterlogged wood.
Yes, I worked also a lot with the archaeological waterlogged wood, since in Ljubljana we have the pile dwelling from Ljubljana. And I developed a protocol for SEM, scanning electron microscope, that we can apply to the waterlogged archaeological wood. It's published, this protocol, and we were able to get thin section from frozen wood, and then we mounted it directly on the steps of the scanning electron microscope using a very small amount of albumin. In this way, when it dried in the oven, the albumin did like a sort of glue that prevent the collapse of the thin section, and then we could observe it under the STEM, and we performed also EDAX then.
Okay.
Yeah, it's challenging, but yeah.
Okay, thanks.
Thank you.
Thank you. Any other comments? If no, then Angela,
Maybe
thank you.
Yes, I have another question, please. When you analyze the tannin, is it the aim of quantification of how much tannin there is in the wood samples, or
Yeah,
is it the quality of the tannins?
Both. I reviewed both qualitative techniques and also quantitative. I mean, the methodology that also allow quantification. Usually, this is done after with the image analysis. Once that you can, identify the tannins, and you can make a threshold that give you then with some image system, you can get the threshold, identifying only the tannins, and then you can get a percentage of tannins that are in your image.
And then that makes it possible also to compare different wood species.
Yes.
Yeah. Okay.
Yes.
Okay. Thanks. Yeah.
Yeah. Thank you.
Thank you. Okay. So I think we don't have more questions. And once again, thank you for your presentation.
Thank you. Sorry, I have to leave you because I have the doctor soon. So sorry, I am sorry.
No problem.
Thank
you
you.
can see the other presentations on YouTube. I think within the week it should be.
Okay. Thank you very much to all. Bye.
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 I think the second presentation will be from Anne van Hilst. Please, the floor is yours.
Yes, thank you. Okay, thank you, Jacob. Today, I'd like to tell you something about my short-term scientific mission called Pit Kilns Excavated and Analyzed. And I will focus more on my experience in my short-term scientific mission rather than the research results. So let me start with a short introduction. I'm an archaeologist and I work at the Vrije Universiteit in Amsterdam as a fieldwork lecturer. And we run an archaeological field school on a site in Veldhoven in the south of the Netherlands. You can see it here on the map. And each year we excavate there with first-year students and some advanced students. And on the site, we find remains of Roman settlement, Iron Age features, evidence of Roman ironworking, and relevant to our coast action, Roman charcoal pit kilns. And because I'm interested in these last two topics, and because I noticed some knowledge gaps, I started a PhD project alongside my job. and my PhD focus on charcoal and iron production in the Maasdamerscheld area during the Roman period. And that's roughly this area on the map. So a part of the Netherlands and a part of Belgium. And the first article of my PhD is about identifying charcoal production of pit kilns in the archaeological record, which fits very well with some of the deliverables of the PoTaRCh Working Group 3. So thanks to the STSM grant, I was able to spend a week in the Archaeobotany Research Laboratory in Ghent. And I worked there very closely with Professor Koen de Vors and Aze Peters. I think many of you will know them. They're present because they're both involved in this COST action. Yeah, and the whole team of the lab welcomed me very warmly, and I'm very grateful for that. And the expertise available in Ghent is not present at my home university. So this visit was very valuable for me and my research. So I had two main goals for this STSM, and these were to receive hands-on training in charcoal identification and to learn how to select good radiocarbon dating samples, and especially in the context of pit kilns. Before I tell more about my visit to Ghent, I want to show you the archaeological context from which our samples came. So very briefly, a pit kiln is a below ground structure used to produce charcoal. And after production, most of the charcoal was removed, but the lowest layer usually stayed behind. This was to avoid mixing the produced charcoal with the surrounding sediment. So, and then over time, the pit filled up again. So archaeologically, we typically find two layers, a lower charcoal layer and an upper backfill layer. I also want to explain that at the site we're going to look at, we only have the bottom parts of the pit kilns left, because the upper part was disturbed by medieval ploughing. And I'll show that later in another slide. So at our field school site in Veldhoven, we regularly find pit kilns. Then each year when we open a new trench, we often discover new ones. But of course, we never know in advance, so it's always exciting. And in the video, you can see the trench being excavated by the machine. And on the freshly exposed surface, where my colleague is carefully skim shoveling to make the surface readable and therefore the features visible, you can already spot a pit kiln. It's the light gray feature over here. So for everybody who finds it hard to see, here are Aze and I pointing the feature out. Aze visited the excavation specifically to look at the pit kilns, and as you can probably see from our faces, we are very happy to find another one. So the pit kiln is rectangular and probably Roman in date. Its edges are a bit unclear because of bioturbation, so animal activity, roots, and other processes that have disturbed the soil. But you can already see a concentration of charcoal. Sorry, I got a message that there is some delay in the slides. Does anybody see a pit kiln with Aze and me?
Yes, I do.
You do?
Yes.
Yeah, for me as well.
And it's a specific, I voted, maybe it's a specific problem with my connection.
Okay. So then I think I'll, okay, sorry. Thank you. So sorry, say again. So you can see a concentration of charcoal on this side, but what we see is backfill of the pit kiln. So here is another example of a pit kiln where the charcoal is much more visible, which makes the identification easier. And again, this is a rectangular Roman pit kiln, though the edges are still also disturbed. And in the video, you can see a colleague creating a section through the feature. And these 3 photos show the same feature. And in the close-up, I hope you can see it, already shows large fragment of charcoal visible in the feature film. And in the middle photo, shows how the shapes becomes clearer as we deepen the section and are entering this charcoal-rich layer. And in the profile, again, disturbed by turbation, you can hopefully still see the 2 layers. So the light gray backfill on top and the remaining charcoal layer below. I think it's maybe a little bit hard to see. You can also see the distinct pit shape, a flat bottom with relatively straight walls. And in the trench wall behind it, over here, I hope that's clear, there are also fragments of charcoal visible. And this shows that only the bottom part of the pit kiln has survived, and the upper part was incorporated in the medieval plagan soil by ploughing. So the charcoal stayed roughly in place, but the boundaries of the features are gone. And yeah, that's why we know we're at least missing 30 centimeters of depth. So I also added these 2 Roman, they're already radiocarbon dated pit kilns from earlier campaigns on the same site, where the edges in profile are much clearer, and the layers, so the remaining charcoal layer and the backfill are easier to see. So we took the entire fill of the features as a sample and wet sieved them, and here you can see part of the charcoal from feature AT. Yeah, not a scale bar, but if you look at the newspapers, big pieces of charcoal are visible after the sieving. And I'm always quite surprised how much charcoal is left behind in the kilns, and also that this is only visible after the sieving. And it's quite hard to see during fieldwork how much it is. So once the sample had dried, I was ready to go to Ghent. So in the lab, Koen and Aze showed me how to identify charcoal under the microscope. And in my PhD research, I work a lot with anthropological data, but I had never actually done any identification myself or saw the lab work of these identifications. So in the videos, Aze is looking at a charcoal fragment again from the feature we just saw. So I learned that charcoal needs to be examined in the 3 different anatomical sections. So a transverse, a radial, and a tangential. I still need to work on my pronunciation on that one. But yeah, we analyzed many fragments, but I will show you two examples today. So this photo we took in the lab shows a transverse section of oak. And oak is very recognizable. I learned it had large early wood pores, smaller late wood pores, a typical flamed palm pattern, and both single here, single race and multi-serrated race. So in the dated pit kilns from the Netherlands and Belgium, oak is by far the most commonly used taxon. And one characteristic of pit kilns is the low taxonomic diversity. So usually charcoal from just one species, and usually oak, with maximum only a few % of another species in the charcoal sample, which was often used as a covering material during production. So during my STSM, I finally saw the oak under the mic, so I was happy with that. And another important part of my visit was learning how to select good samples for radiocarbon dating. Because oak is a long-lived taxon, the old wood effect in radiocarbon dating is a real problem. If you date older hardwood, the results can be 100 years older than the actual charcoal production event, which is what you want to date. So this issue of the old wood effect is a problem in archaeology in general, but for pit kilns, the error can be especially large because all trees from long-living species were used for charcoal production. That's why for dating this pit kiln, the next fragment is so much better. And I was happy we found it in the sample. So this is also a photo taken in the lab, and it's a fragment of Cytisus, broom. The pore pattern and the ray width are very different than oak. It's maybe a little bit hard to see, and I think under the mic it shows much more clearer than on the photo. But yeah, broom is a short-lived scrub, which makes it ideal for radiocarbon dating because of the risk of the old wood effect is almost non-existent. And broom is not chosen to produce charcoal from, because the charcoal wouldn't be of any good quality from the scrub. So therefore, we expect it to be a covering material used to build the pit kiln. So thanks to this short-term scientific mission, I gained some practical skills and now understand charcoal identification and radiocarbon sample selection much better. And I improved my ability to interpret archaeological charcoal assemblages in relation to charcoal production. And besides the lab work, I met the whole arboreal team, which was very lovely. And I received help with R analysis, which was perfect timing for my research. I joined a project meeting where Asa presented her research to a multidisciplinary panel, which was very interesting and motivating, and discussed our pit kiln article with Professor Koen de Vors, which made the progress so much better. So I really want to thanks everybody involved in making this short-term scientific mission possible, and thank you all today for listening to my experience.
Thank you for the presentation. I really like the videos from the excavation. As I'm not an archaeologist, it's super interesting to see how it actually works. And then how you use the wood science to combined with archaeology. Really interesting. And any questions, any comments? Yes, Oliver.
I have a question. Thank you for this presentation, very nice. And sorry for I had some technical problem with the delay of the slides. My question is about the stratigraphy of the pits. You said you had the bottom, like the bottom stratum, the bottom layer from the actual using the pit, and then the infill.
Yes.
Do you have any evidence at some of these pits? I mean, it's difficult because it's so this dominance of oak, so you don't have evidence from the species. But is there any evidence that you have like two usage layers or different like pits were probably used several times? So do you have any stratigraphic evidence for this, or is it not possible to say?
I don't know if I understand your question totally, but we do find pit kilns where we see multiple charcoal layers left behind. Yes.
Okay. Yeah.
So
okay. You have that.
but they're not very common, and I think in the production during production, it's also very convenient to empty the pit again at almost the same depth and the same width of the. So that's why we can't see those layers all the time. So so most of the time we can't see if a pit kiln is being reused, but we have a handful of examples where it is visible.
Okay, fine. Yeah. Thanks.
Maybe Oliver, I'd like to add to this that I think in fact it's very likely that these pits have been reused, because when you have a complete cycle of charcoal production in the kiln, you extract the charcoal and you end up with a pit. And the volumes of charcoal produced in one go in one charcoal kiln, it's quite small. So there was probably a lot of wood left in the vegetation. And then if you have a pit that you can, it makes sense to reuse the pit instead of making a new one. And so I think these have been reused probably regularly, but I think in a very short period of time. Because it's all these pits, well, at least in Belgium and the Netherlands, they're in very loose sediment, in sand or loam. And if you leave them like this, they backfill by themselves. And so I think we should be aware that there was probably multiple uses, but we can consider it as a single event, because in the meantime, the vegetation will not have changed. I think this reuse was within days, maximum weeks.
Yeah, I see.
And so in an archaeological point of view, or for paracological reconstructions, I think we can consider it as a one moment in the past.
Yeah, makes sense. Yeah, thanks.
Thank you. Any more comments? If no, we can proceed with the third presentation.
Maybe one question.
Yes, sure.
Do you have any idea of the diameter of the wood which was used in these pits? Big trunks or also branches?
Well, so this is all I got from the specialist data, but what I read about it is that also very big and whole trunks were used. And there is also research in the Netherlands where they look at the tree ring division between a part of charcoal, and they see it's part of really big stems and really old stems. Okay. And what we also see is that because of this old wood effect, that we have, if we take multiple samples, radiocarbon samples from one pit kiln, that sometimes there is a really big difference between the correct dates, the younger dates done on which are the result of the right sample choosing, and the older dates from probably like parts of the hardwood, and there also can be like more than 100 years in between those dates.
Okay. Yeah, yeah, interesting.
Yes,
Okay,
that's really nice. Yeah.
Okay.
Maybe I can add something as well there. I can confirm what Anna said, and it's even to the extent that when we were looking for radiocarbon samples, we prefer twigs, and we often have to do a lot of analysis to end up with maybe one fragment of a twig, but then in a lot of cases we don't have any. So that's good indication as well that they used quite large diameter of woods. Often you don't find any small diameter woods in these older kilns. This is for the Roman period and early medieval period. If in the post medieval period, it's another story. But in these pit kilns, it's we don't have any small diameter wood in general.
Okay. And then more comments? If no, then our third presentation by Mehmet Ergun.
Okay. Hello, everyone. Can see some old friends and hello, other new friends. Thank you very much. My name is Mehmet Emin Ergun. Firstly, I can share my presentation. Can you see my presentation?
Yes.
Perfect. Firstly, I introduce myself. My name is Mehmet Emin Ergun. I'm an associate professor at Alanya KOH University in Turkey. My presentation title is Preparation and Characterization of Activated Carbon from Waste Wood for use in the wood-based panel industry. Maybe you know maybe you see the news in 2021. We have a severe forest fire in Turkey during the summer of 2021, permanently changed our country's landscape. They burnt over 200,000 hectares of forest and caused enormous ecological and financial damage. And total 183 different areas had forest fire in 2021 summer, especially southwest of Turkey had a severe damage to forest fire. As you can see, the red color in Turkey in this map, it's out of Pinus brutia red pine grow. The red pine has formed extensive pure forests on the coastal slopes of the Mediterranean, Asian, and Marmara regions, and it can grow at altitudes of up to 1,500 meters. It is most abundant in the southwestern part of the Turkey. A portion of the wood was harvested and sold to meet economic needs and market demands, but unfortunately, a significant amount was left to decompose in the forest. Furthermore, even large diameter logs and branch wood were used only for heating purposes. Also, these last two pictures show this wood provided by supported to our school for heating purposes to our students, and all of this wood is also red pine. Our main objective is to produce activated carbon from fire-damaged red pine wood waste and utilize it as an additive in particleboard manufacturers. We want and expect it to dual benefits. First one is environmental benefits contributing to circular economy by valorizing forest wood waste. The industrial side, reducing the formaldehyde emission from particle boards to create healthier products, because you know formaldehyde molecules is carcinogenic effect and it leads to respiration problem. And contribution to this cost action, a traditional forest byproduct has been characterized using modern methods, thereby supporting these actions objectives. It's a lot of work in this study, so I have divided in two sections. First one during the STSM, the second section is after the STSM. The first section is during STSM in Poznań University of Life Science. We produce activated carbon production and make some characterization. Material and methods. Firstly, red pine wood waste placed in the oven for carbonization at 600 Celsius for 1 hour with oxygen-free condition. After we obtain a biochar, for chemical activation, we use the two chemical activation. First one is potassium hydroxide. The carbonized red pine wood was mixed and grounded with potassium hydroxide. And the second one is carbonized wood was mixed and grounded with potassium carbonate. After the activation temperature was 750 Celsius in tube oven for 15 minutes. After the activation, the activated carbon was extracted in a succinate device for 8 hours using 2% hydrochloric acid, and then another 8 hours with distilled water for 2 times. After we dry in the oven for 1 night and grinding, and it's ready for the use for particleboard production. Characterization of activated carbon. Firstly, as you can see the image, the left side is the microstructure of the potassium hydroxide activated carbon appears more smaller and homogeneous structure compared to the other one. On the other hand, the structure of the potassium carbonate activated carbon is more heterogeneous and with larger particle size. Soon in the batch results, we will see similar results regarding the pore diameters of this activated carbon. And we are looking at the XRD pattern of activated carbon and wood. Firstly, the green pattern is wood. The distinct peak around approximately 16 and 22 observed from the crystalline region of cellulose. This pattern indicates lignocellulosic structure of the wood. On the other hand, both chemical activation methods successfully disrupted the crystalline structure of the wood. The more aggressive effect of potassium hydroxide create a more amorphous carbon structure, which is associated with a higher surface area. In contrast, potassium carbonates less destructive effect. Then we are looking at the elemental analysis. The carbon content of the raw wood material, 50% after the chemical treatment, increased to 70% in potassium carbonate activated carbon and 84% in the potassium hydroxide activated carbon. Conversely, the oxygen and hydrogen content, which were high in raw wood, significantly decreased. The oxygen content is decreased, particularly more pronounced in the potassium hydroxide activated sample, because as I said before, potassium hydroxide is much more effective and aggressive than potassium carbonate. And similarly, the hydrogen content of potassium hydroxide activates carbon higher decrease rate than other activated carbon. It means potassium hydroxide activated carbon has a comprehensive breakdown of aliphatic components and the formation of a more developed carbon network. For properties and yield of activated carbon, the surface area of potassium hydroxide activated carbon is 1,953, while the surface area of potassium carbonate activated carbon is 568. Furthermore, both activated carbon have a predominantly microporous structure, as you can see. But when we are looking at the yield of activated carbon, the potassium carbonate activated carbon should have been kept at 2 ohm for a longer duration, because this percentage is high. I think we expect a little bit lower. Determination of surface functional group and iodine number. Surface chemistry analysis show that the potassium hydroxide activated carbon contains greater amounts of both acidic and basic functional group than the other activated carbon sample. This surface group enhance the potential absorption of polar compounds. It make more beneficial for the absorption applications. The iodine number results, which reflect the microporous absorption capacity, show a strong dependence on the activation agent. The potassium hydroxide activated carbon showed a significantly higher iodine number compared to other activated carbon. It's correlates with its larger micropore volume and higher surface area, as we can see the wet results. And we have a lot of work in the study, so it's not possible to finish just 10 days. So I continued in Barton University. After STSM in Barton University, we produced particle boards and characterization some tests. Firstly, before the adhesive treatment, the chips were dried 1 to 3% moisture content, and chips were bonded using formaldehyde adhesive, modified with 1%, 2.5, and 4% activated carbon based on the dry weight of the adhesive. The mixture were mixed using a mechanical stirrer, and the adhesive was applied to the chips using an adhesive gun within rotary drum mixer. And as you can see, the glue chip for the layer, and after the manual matte formation process using a wooden spreading mold, after we applied the cold press to the matte formation, and the formed mats are pressed under the hot press. The particle board mats were hot pressed for 4 minutes at 180 Celsius. And as you can see, the single layer particle board. This is two different types of particle board. Left side is particle board without activated carbon. Right side is particle boards have activated carbon. As you can see, the black spot on the particle board surface is activated carbon. It's distributing the particle board surface. I want to show you. And characterization of particle board reinforced with activated carbon. firstly, we may the physical properties of particle board. The addition of activated carbon to the particle board enhanced in thickness, swelling, and water absorption values compared the control group. As the amount of activated carbon increased, the thickness, swelling, and water absorption values decreased. This situation, I think, related to we saw from elemental analysis results, the amount of hydrogen and oxygen in the activated carbon decreased. Fiber formed fewer bonds with water. The activated carbon coated the surface, reducing the water absorption by preventing the interaction between water and fiber. And mechanical properties of particle board. Mechanical properties of particle board improved with a special 1% additional activated carbon. However, with the addition of 2.5% and 4%, a decrease in a mechanical properties was observed because it reduced the interaction between the fiber and the adhesive, and activated carbon blocked the fiber-fiber interactions. And free formaldehyde emission of particle board. When we are looking at the results, the formaldehyde emission values, a reduction is observed in both groups compared to control groups. And the formaldehyde emission decreased as the amount of the activated carbon increases. A greater reduction is seen in the particle board containing activated carbon produced with potassium hydroxide. This situation is related to the higher surface area of activated carbon produced with potassium hydroxide. And we are looking inside the particle board. The activated carbon absorbs the formaldehyde. The pore structure of activated carbon is crucial for its absorption performance. Micropores, which are smaller than 2 nanometers, it's directly related to absorb small molecules such as gas and organic contaminants. Mesopores, ranging from 2 to 50 nanometers, facilitate the transport of larger molecules, thereby enhancing the diffusion and overall absorption capacity. And macro pore upper from the 50 nanometers. And when we are looking at the absorption mechanism, microstructure of activated carbon directly reduces the formaldehyde emission through absorption. Its polar surface compound and oxygen-rich functional groups capture formaldehyde molecules with hydrogen bonding and van der Waals forces. And conclusion, two different types of activated carbon were successfully produced from fire damaged red pine wood waste. Potassium hydroxide activated carbon exhibit a significantly higher surface area than the other activated carbon. The addition of activated carbon improved the mechanical properties and water resistance of the particle board, and activated carbon incorporation also contributed to a reduction in formaldehyde emission. And this study presents a sustainable model supporting the circular economy. And we will finish writing the article in 2 weeks. I know it's important to you as well as important to us. And we emphasize your support. Don't worry about that. And I would like to express my sincere gratitude to Professor Beata Dostekowska for her excellent hosting and supervising during my study, because it's my first international collaboration. Thank you very much. And also thank you very much to her wonderful research team, all of them very kind and helpful to me. Our collaboration is still continuing. And thank you very much for Turkish colleagues in Barton University. And this study was supported and carried out within the scope of this STSM under this cost action. And thank you very much for listening. If you have any question, please send me an email, and thank you.
Thank you for the presentation. And I'm very happy that my department could help you with your research. And that you so quickly wrote the article, which
approximately before the end of this year we can send somewhere.
Great.
I'm quick always.
Thank you. And any questions or comments?
No.
Yes, maybe one short question, Mehmet.
Yes.
Thank you for your presentation. What's the percentage of the activated carbon in the particle boards? Maybe I've missed that, because on the picture you showed, they were quite tiny spots.
No problem. I can show again. Could you see?
Yes.
My presentation. Sorry. 1%, 2.5, and 4%. It's
okay.
Thank you.
Yeah, thank you. I missed that slide. Sorry.
No problem. I'm quick because when I was first time in Poznań, I fired in the stage. I don't want again.
No, today we had more time. Yeah. So, if there are no additional questions, you can always write an email with the questions to Mehmet and to other presenters to ask about the details. Also, don't forget that soon we will announce another batch of STSMs and grants. The meeting of the core group is in Rome in 2 weeks or 3 weeks, and then we will start to announce what will happen in the next grant period, and then the details of where, at what you can apply and what the dates of the applying. So the new grant period will start soon also. So you are welcome to use our support, and we are happy to help you. So thank you for today, and see you.
Thank you very much.
Jacob.
Thank you, and see you. Yeah. Bye-bye.
Bye.